Hello, I'm Yan Fukuo from National Taiwan University, and it is my honor to be invited to this NARO-FFTC event to present my research. And the title of my research I'm going to talk about today is Identifying Tomato Diseases and Pests Using Intelligent Machine Vision. And in this research, we developed a line bot over here. You can see the friend ID or the QR code. You can add this line bot as a friend. So the research is about tomato, and we know tomato is a very important crop over the world. And over the world, you can see the production area, the production every year, and also their revenue. And in Taiwan, it is an important crop too. But although it is important that we know disease can cause the loss in the crop, and if we don't do anything, the loss can be very serious. And disease identification is very important as a part of the IPM, the integrated pest management. The identification is here. And this is because we want to use the correct pesticide. We don't want to use the wrong pesticide that can contaminate the environment. So how do we identify the disease? We basically rely on the experts, experienced farmers, or plant pathologists, but they are not always there available. And if there are too many diseases needs to be identified, the pathologist can be overwhelmed too. So here we want to develop a solution for identifying the diseases in the field. And this solution needs to be fully automatic, and it needs to have high accuracy, and also it has to provide the service 24/7. And before we start, we have to realize something. So first, in the field, we don't have culture or microscopy that can help us. The identification of the disease mainly depends on the naked eye. So if we want to use AI to identify the disease, we have to remember this. So here are the disease images that we collect in the field. Totally, we have 11 disease and pests. But we don't want to identify, or I should say, we cannot identify the 11 disease. That is because some disease in their early stage, they look very similar. So here, for example, we have bacteria spot, and we have gray leaf spot, and we have early blight, and the target spot. If they are in their early stage, they look very similar. And even for the plant pathologists, they cannot identify the disease by themselves. So actually, in this research, we group them together. Here, this is called one category. Okay, and the same thing happened on early blight, the late stage, and also the target spot, the late stage. They are another category. 2. So totally we have 1, 2, 3, 4, 5, 6, 7, 8. 8 categories. We put the disease, or we group the disease together, and we want to identify the group only. So here are the 8 categories. And the total solution look like this. Here we have a cell phone of the farmers or the plant pathologist. And the cell phone can take an image of the leaf with disease lesions. And the farmers can upload this image through the line Messenger, a software that is commonly used in Taiwan as well as in Japan. And this line, we call it line bot, can communicate with three models, three deep learning models on the cloud. That is a server, a cloud server in our lab. And the server has three deep learning models. You can see 1, 2, 3 over here. And the communication between LiveBot and the three models are depends on this relies on this chatbot controller over here. Okay, and what are the three models used for? The first model is called ADM. It's anomaly detection model. The second model is disease identification model. The third model is leaf mold powdery mildew 2 distinguished model. I will explain them separately here. So let's check the anomaly model. And we know in the field, when the farmers take a picture, they don't necessarily take the picture as the picture you want. They may take picture of this like you can see there's a bush over there and there's some clothes over there too. This is not the image that we want. So we call this anonymous image. What we want is an image that contains a leaf over here. And this is called normal image. And that's the purpose of this anomaly detection model. The model wants to exclude this image that we don't want. And this is the architecture of the model. We have a feature extractor in this ADM model, and it's VGG11. It's a deep learning model. And we also have an isolation forest that is attached after the VGG11, and that is used to distinguish those anomalous images. And we collect a total of 300 plus normal images. And also they are anomalous images that we download from the internet. And you can see some example image over here. And this is the classification results. In this ADM classification, we want to distinguish normal from anomalous. And based on our test results, that average accuracy is 97.4%. Okay, and let's move to the next model. That's the disease identification model. And this is the major contribution of our research. And the disease identification model is basically a YOLO V4 model. And it's a object detection and classification model. In a sense, if we provide image of tomato leaf, and you can see there's a lesion over here, and we feed this image into the YOLO V4 model, the model will determine the location of the lesion and tell us what kind of lesion it is. And in this image, in this example, it is yellow spots. So we collect the image of the 8 categories, okay, and they look like this. And you can see in this image that the leaf area is actually pretty big. It's like 1/3 to 1/2 that large. So the symptom or the lesions are obvious. And here are the amount of images that we collect. We collect a total that is almost nearly 9,000 images. And we also have to annotate the bounding box. That is the lesion of the on the leaves. And we actually annotate a total of 14,000 lesions. And before we train the images, the models, we also do some image augmentation. And the augmentation includes saturation, exposure, hue, and mosaic. And here is the performance of the disease identification model. Here you can see that it reached the mean average precision MAP of approximately 76%. It doesn't look high, but let's check it later. Here are the results. You can see that under the circumstance that the lesion is very light, see it's only a little bit wide, and we can still detect it. And for the case that the image background is very complicated, you see this is the leaf, and the backgrounds is very complicated. We can still detect it, and we can detect several objects in one image. So here are our achievement. And also in the case that in one image there are different types of categories or lesions or diseases, we can detect them too. See here we have powdery mildew, and we have yellow, and here we have water soak and leaf miner. They are all different diseases. But there are still some failure cases, but failure is not actually that failure. I should explain it here. See, we have sometimes the ground truth bounding box, that green one, is very big, but the detections are small. But the small bounding box, they indeed enclose the lesions. But actually, in the object detection, this is considered a failure. This is because the IOU, the intersection over union, is actually very small. It's smaller than 0.6. So these two images are considered failure cases. So in order to prevent this issue, we want to convert the object detection problem to a identification or say classification problem. In that sense, as long as we detect a lesion, and no matter how big the lesion area is, it should be considered a successful detection. So here we do some manipulation. We want to make sure that if the image is one-to-one mapping, or it is multiple to multiple mapping, or it is no detection. And there's some conversion, and after that we convert the MAP, that is approximately 76%, to the accuracy, detection accuracy, that is 93%. So this is a much better results. Okay, so let's move to the third model. That is the leaf mold powdery mildew 2 distinguished model. And in this model, it overcomes the issue that on the upper side of leaf, that these two diseases, they look identical. But on the lower side of leaf, we can tell their differences. So this model, I should say, if we detect that disease is either powdery mildew 2 or is leaf mold, the live chatbot will ask the farmer to turn over the leaf and ask for an image of the lower side of the leaf, and we do another classification. So this is how this LPDM work. So here are the training images, and this is the model that we use on this LPDM, and here we can see the results. The accuracy is 98%. And here is a video that shows how this chatbot works. So first thing, you have to upload an image to chatbot. You can either turn on your camera or select image from your camera rolls. And after you submit one image, it will return this message that says that disease are identified. And you can click the image and it will enlarge the image. And here that you can see if there's a disease identified, here are the treatment or possible disease explained as a hyperlink. And so if you click this treatment, or it will tell you more details. So this is basically my presentation, and here are a very short conclusion that summarize the accuracy of the models. And before I end this presentation, I would like to thank the Ministry of Science and Technology of Taiwan for their sponsor of this project. And this research cannot be done with the partner in the Plant Pathology Division of TARI. Thank you very much for your attention.